Good. So this is a nerd out session, and I'm really glad and grateful to have an opportunity to talk about the history of water quality metrics, not in the sense that because it helps people apply the current metrics by understanding some of the history. We understand where they come from, and we understand some of the benefits and limitations of the current metrics. So I'm really happy to be able to go through these. My personal history was that I worked with recreational water for about 15 years, and I was based in and around Cincinnati, Ohio, where some of the premier scientists at EPA who developed some of the standards were working. I actually got to work personally with some of those individuals. So this is one of the reasons why I was able to obtain a historical perspective that I think is worth sharing. And we won't share the entire thing. We'll just share some of the things that I think that may be valuable to you as you try to understand where do these numbers come from, what do they mean, and what's the appropriate way to interpret things, or what's a useful way to interpret things. So first we have to go through just a little bit about produce safety rule compliance dates because we want to ensure that there is no confusion. Then we'll talk about the history of the metrics and how the development of the field of microbiology led to improvements in the quality metrics that have been used over time and how they're linked together. We'll talk a little bit about the epidemiological data. In other words, the data that show at this level, this many people get sick, and that helps to establish what are the appropriate levels for water quality standards. And then just briefly, we'll talk about CDFA produce safety program and produce safety rule inspections, just for your awareness in case you weren't part of the other two sessions. So starting with a reminder that produce safety rule compliance dates are currently understood to start no earlier than January of 2022. And we are still, as Linda mentioned, we're still awaiting the revised produce safety rule requirements for Subpart E, and which ultimately will come into play if you are a farm covered by the produce safety rule. In the meantime, a farm is still prohibited from selling adulterated produce. And we'll talk about that in more depth and why that word is so important to you. But I just wanted to put it out there. We're not going to read through all these. You can take a screenshot if you didn't see the other sections, or check our produce safety program webpage at CDFA, or the UC Davis has produce safety information on their website. Leafy Greens Marketing Agreement, technically the California Leafy Greens Products Handler Marketing Agreement, is another set of rules and metrics that began in about 2007, partly in response to that outbreak, a famous outbreak related to spinach consumption. The causative agent was E. coli 157, which is one of the pathogenic types of E. coli. So LGMA is centered in California. It's centered on spinach, lettuce, and other leafy greens. There's a sister organization in Arizona. And just to clarify, there are audits associated with LGMA membership, and those are performed through CDFA, but that's a different program from the produce safety program. So just wanted to get that on the table. What's important about these requirements for water quality, whether they be FSMA, produce safety rule, or LGMA, is that they're based on good agricultural practices. And in our previous two sessions, and Ronnie later this morning, deal with some of the good agricultural practices, usually abbreviated as GAPs. Farms must inspect an agricultural water system. That's part of both produce safety rule and LGMA. The goal is to identify hazards to water quality. Typically, these are fecal contamination related hazards, because most of the produce-borne outbreaks tend to be fecal oral pathogens, the pathogens that originate with feces and unfortunately get ingested. They get consumed by mouth, that's the oral part, and cause diarrhea disease in the person who's unfortunate enough to eat the contaminated product. Ensuring the system is in good repair is another part of the inspection of the ag water system. The other good agricultural practice, the other GAPS practice, is routine testing throughout the growing season and quantitative analysis for generic E. coli for irrigation water purposes. Farms must maintain quality of their water, and then the federal requirement that adulterated produce may not be sold as food, which I'm going to use as a segue into a conversation about why are we testing our water for E. coli? Why are we testing for fecal contamination? The federal code is that a food is adulterated if, and there are 7 different conditions in the full definition, but I want you to focus on the ones that are in red here. If that food consists wholly or in part of filthy, putrid, decomposed substances, I wouldn't want to eat it. If it's been prepared, packed, or held under insanitary conditions where it might become filthy, putrid, or decomposed, or contaminated with filth, I should say, well, those are both part of the definition. Those are both conditions. Those are all conditions for adulteration. In other words, a product that is not fit to be eaten as food because it has a health risk. So as a producer, as a grower, as a farmer, how would you know that your produce had become adulterated through use of water that's of insufficient quality? So the gaps involve both inspection and analysis. What we want to talk about in this webinar to bring us around to the point of why are we even talking about this is that sanitary quality of water is hard to predict based on past lab analysis. And we don't say this later in the webinar, so I'll say it now. When a farm sends a water sample to the laboratory, it's going to take anywhere from 2 days to a couple of weeks for the result to come back. And by the time the result comes back, the quality of the water that they're using may have already changed. So it's virtually impossible to know the quality of the water at the time that you are using the water, because the laboratory analysis that's available today takes time. It takes at least 24 hours, and then there's some lag time before the results get released. So the take-home message from this webinar is it's a mixed message. What we're hoping to do is underscore the fact that the E. coli test is a useful indication of fecal contamination. And feces is generally considered to be filthy and insanitary, which relates to the definition of adulteration. But then the key question is it a little bit of feces or is it a lot of feces? How much fecal contamination is too much? How much does it take to actually be considered adulterated? And the Department of Public Health, when they make a determination of adulteration, they typically are going to want to see that there are actually pathogens on the produce. So at what point do you get to the point where there are actually pathogens on the produce? And the E. coli number, and that's going to be the whole point of this next 20 minutes, is the E. coli number by itself does not necessarily predict safe or unsafe status or whether or not it's going to put pathogens on the produce. It is great to identify whether there's poop in the water or not. If you had the choice, I would prefer that we use water that doesn't have poop in it compared with water that doesn't have poop in it. Wait, you know what I mean. We want to use water that's free of fecal contamination. But knowing that surface water is vulnerable to some level of fecal contamination, it's very difficult to ascertain, to determine how much is too much. And the E. coli test has some limitations, and that's what we're going to talk about. Hugely valuable to do the analysis and to look at the trends, but the number by itself is not a magic cutoff line that people sometimes rely on. So let's get past that. This is a dense slide, and I'm not going to go through these requirements of the Produce Safety Rule and LGMA. They're complicated. They are set out in multi-page documents. But if anybody grabs a screenshot, I wanted you to have all the information in one spot, even if it is complex, and we're not going to take the time to describe the entire microbial water quality profile system and the type A, type B system, LGMA. What I want you to focus on is that there are these numbers. And I referred earlier to magic numbers. 126, 410, no detect, which is comparable to drinking water, 126, 576. These are numbers that when a person who doesn't understand, a person who's not aware of the background, if you see a number that says, okay, if it's normally 126 or less, it is the Produce Safety Rule as written would allow for it to be used in these purposes. And it's easy to jump to the conclusion that less than 26 is safe, more than 126 is not safe. And that is not necessarily the case in this talk. We'll describe why. So in order to get there, this is where the geek out starts, if it hasn't started already. We'll go back 100 years to 1922. In 1922, the United States and the rest of the world had just recovered from tremendous pandemics. We had Spanish influenza. Well, it wasn't really originated in Spain, but let's call it the influenza pandemic of 1917, 1918. We had cholera, we had tuberculosis, we had leprosy, we had typhoid fever. If anybody remembers the story of Typhoid Mary spreading typhoid fever. Diseases were not under control in the way that they are in this current generation. I mean, all of those diseases that I just mentioned in at least a localized way were as impactful as COVID-19 has been in the past year. I'll throw that idea out there that tremendous numbers of people were dying of disease that are currently preventable diseases because of advances in monitoring, in medicine, and in sanitation engineering. But in 1922, it was still common practice for a city to discharge their raw sewage into waterways. And the American Public Health Association and scientists of the day understood that there was a link between fecal contamination in the water and people who were getting sick from swimming in the water. What they were challenged by was, well, how do you know if the water is unsafe and not fit for swimming? And because there was no metric, there was no measurement that could be made at the time. The APHA decided at that time that there wasn't enough science to support any standards because there was no epidemiological data. There was no linkage between water quality metric measurements and how many people got sick. So any standards they created were going to be arbitrary. By 1963, though, 38 states, more than half the states, had taken the initiative, and they had established bathing beach standards between 50 and 2,400 total coliforms in 100 mils. About half of them had settled on the nice round number of 1,000. So we'll talk a little bit more about where that 1,000 came from after I talk a little bit about the science. At the time, 1963, the total coliform test was about the only routine test for wastewater contamination. There were a couple of common methods that had just great names: lauryl tryptose broth, brilliant green bile lactose broth, named after the pH indicator that was in BGBLB. Great informative names. What they did was they selected for total coliforms, and I'm going to go to this slide first. A total coliform is functionally anything that grows in those media with characteristic results. A total coliform is going to be gram negative. It's going to ferment lactose, which means that it'll lower the pH and create carbon dioxide. BGBLB starts brilliant green, as you might have expected. And when the pH drops, it turns acidic and it turns yellow, and the gas formation can be determined, can be seen because gas is collected in a Durham tube. This is very analog. I mean, this is 1963. This is direct observation of what can grow on lactose at 35 degrees, creating gas and lowering the pH, or on M-endo, it's going to be metallic sheen. So that's a functional definition. So total coliforms is the full group of organisms that have those characteristics. When we get a little further, we'll talk about fecal coliforms and E. coli, because the follow-on tests were able to detect more and more narrowly fecal coliforms than E. coli. And we'll talk about those as we get to that evolution in the process. But what I want to make sure that we understand from this diagram is that fecal coliforms are a type of total coliform. E. coli is a type of fecal coliform, and most of the pathogens aren't coliforms at all. So in other words, these tests are going to tell you whether the feces is present, but they're not going to directly tell you whether these fecal oral pathogens are present. So where did the 1,000 coliforms per 100 mils come from? Well, 100 milliliters is about 4 ounces. It's about a half a cup, and that I should have double checked this. That's about the amount of water that is ingested during swimming in a normal swimming event. So 1,000 coliforms that a person is going to accidentally ingest while swimming. So how did they arrive at 1,000? Well, I think the most important one was by the Ohio River Valley Sanitation Commission. The Ohio River Valley, specifically Cincinnati and Pittsburgh, were major dischargers of raw sewage to the Ohio River. And that's one of the reasons why Cincinnati was such a hub, because Cincinnati is right there on the river and was dealing with the water quality issues centrally located. The Ohio River Sanitation Commission, this is before EPA was in Cincinnati, measured both the pathogen S. typhosa, which is not the current name of it, but that was a major cause of diarrheal disease. And they measured total coliforms, and they concluded that at 1,000 coliforms per 100 mils, they very rarely found even one cell of S. typhosa. So they thought, okay, 1,000 coliforms is going to be protective, because at that level, above that level, we're more commonly finding S. typhosa. Below that level, we're rarely finding S. typhosa. It's going to be protective of health. In Connecticut, they did a survey of the coastline, and they found that 90% of the time they were hitting that 1,000 coliforms, total coliforms limit. So what does that mean? That means it's achievable. It's not going to hurt that much to incorporate this 1,000 total coliforms limit. And then California Bureau of Sanitary Engineering in 1943 came out with a decision, a blue ribbon panel report that said, okay, at 1,000 total coliforms, the current standard, the contemporary standard for drinking water was 2 coliforms. So it's about 500 times safer than drinking water. Yeah, I can go along with that. If it's 500, if it can be 500 times more than drinking water and still be safe enough to swim in. And so you can see that there's a combination of judgment and science and practicality that went behind that limit. It needs to be the limits that were being established for standards 1963, 50 years ago, 60 years ago. They need to be achievable. They need to be protective of public health, and they need to be consistent with other standards, at least by a gut check. Total coliforms is not a great indicator of fecal contamination, because you will also find total coliforms everywhere in the surface environment. Total coliforms are on leaves. Total coliforms are in soil. Total coliforms are naturally in the water. They're not a specific indicator of fecal contamination. So the U.S. Public Health Service did 3 geographic areas. Again, you'll see Cincinnati in the list because Cincinnati was a hub for this kind of work at that time. But they also hit Long Island Sound and Chicago, because you have major sources of fecal contamination in all 3 of these locations. Because at the time, we were very rarely, if ever, treating our discharged sewage to the waterways. They concluded that they did follow-on epidemiological studies. They concluded that, yeah, 1,000 was conservative. They were able to detect more people get sick above about 2,300 per 100 mils of total coliforms. But they weren't happy with total coliforms, because total coliforms are ubiquitous. They're always found in the environment. So they said, well, let's use fecal coliforms. And they did the same studies, and they showed that the action level, the point at which health effects were detectable, was 400 per 100 mils. So they recommended that normally half the action level, and no more than 10% of the time higher than the action level. So very rarely will you be at the point where people are actually going to get sick from swimming in there. So what's a fecal coliform? A fecal coliform is nothing but a total coliform that grows at 44.5 degrees instead of 37 degrees. In other words, it's above body temperature. They also consistently include some bile salts. Bile salts are similar to what's in the human gastrointestinal tract, so that's another selective agent for fecal coliforms. That fecal coliform standard was protective, the same level of achievability as total coliforms, because it's indexed to the same numbers, and it's consistent with that total coliform standard otherwise. The common methods, just for curiosity, the same methods as coliforms, but incubating at 44.5 degrees, or there was a new method for membrane filtration called MFC agar, membrane filtration fecal coliform agar, that was used to detect those, for what it's worth. Then in the 1970s, we go from fecal coliforms to E. coli. We finally get to something that is currently relevant, but this lineage goes back, and we're linking all the way back to the 1920s and 1950s with this lineage of numbers. US EPA was formed in the early 70s. one of the earliest initiatives of the newly formed EPA was to do epidemiological studies on marine and freshwater beaches. They wanted to resolve this issue. What's the best indicator? They had some contenders: total coliforms, fecal coliforms, because they had been used. Then there was a newcomer to the pack called E. coli, which you've all heard of because we already talked about it this morning, and also enterococci. They published some recommendations that were indexed to the fecal coliform numbers, which were indexed to the total coliform numbers. And these numbers are going to sound familiar because I've just been talking about them. 126 is the geometric mean. 235, not currently used, but it used to be used by LGMA. 406 is awfully similar to 410, which is part of the produce safety rule. 576 currently used by LGMA. The calculations were directly related to fecal coliform illness risk levels. And we'll talk about the data behind it after I talk a little bit about the methods. And there were some new methods that allowed us to finally get from fecal coliforms to E. coli. Fecal coliforms are found in the environment more often than E. coli are. And E. coli is considered to be a much more direct and reliable indicator of fecal contamination specifically. Fecal coliforms can still be found outside of feces. E. coli is rarely found other than in feces. So that's why it's a better indicator. So EPA was new. They had all these new tests that were coming out, but they were still going back. M-endo is the total coliform test. One thing that most E. coli have that other total coliforms don't is an enzyme that takes something called MUG, breaks it into two pieces, and one of the pieces fluoresces. So this is why so many of the tests on the market are two-part tests. They start with the total coliform, and then they transfer that in the old style way. They transferred the filter that has all the colonies that were total coliforms onto a different medium that includes MUG, which I can't pronounce what that's an abbreviation for, and you don't care anyway. But MUG then incubates for a couple more hours, it fluoresces, and you can tell which ones of the total coliforms are E. coli. In the 1980s, there was a new auger. It was not a great auger, but it was a new auger called MTEC. In the 2000s, modified MTEC is an improvement on the original MTEC. And then there's another one called, and both MTEC and modified MTEC only detect E. coli. But there is another medium, MI, that does simultaneous detection of both total coliforms and E. coli, and that's used mostly in drinking water matrices. But let's finally get to the data behind it. This is one of the first comprehensive data sets that was generated, and you'll notice the dates: 1979, 1980, 1982, Lake Erie beaches in Ohio and I think Pennsylvania, and then Keystone Lake is I believe in Pennsylvania. So it's a limited geographic area, freshwater bodies. And what they did was they looked at the E. coli density on the day that somebody was swimming, and this is on a log scale, so it goes from 10 to 100 to 1,000 CFU per 100 milliliters. And then how many people at the end, after a week after they swam, how many people reported having diarrhea? How many people reported having gastrointestinal illness or highly credible gastrointestinal illness, because it's not just these bacteria that could give you diarrhea, but if it comes along with a fever, it was considered highly credible. So swimmers or non-swimmers, they almost 8 out of 1,000 people reported having diarrhea after they swam, or I'm sorry, after a visit to the beach. But this was 8 per 1,000 was the background level. And when you get above 126 E. coli, that's when you get above the background levels. And that's where that 126 number came from. And from there, they did some confidence intervals that got us to the numbers of 235, 406, and 576 that were reported out in this same data set. So in 1986, the recommendation was 126 was the geometric mean, and rarely, if ever, it should never exceed 406. In 2012, And that was based on an illness rate of 8 per 1,000 swimmers. In 2012, EPA spent a lot of effort reevaluating those standards, but they came up with essentially the same numbers: 126 geometric mean, 410. The biggest difference was they backed off of the highly credible gastrointestinal, which is this line, and they went to the total GI, which is this line. And in the reanalysis, they come up with an illness rate of 36 individuals who would become sick out of 1,000 who went swimming on that day. So let's finish the story of recreational water by saying that, just making the point that for purposes of recreational water, these numbers were being developed based on science and data since 1922. The current recommendations came out in 1986. By 1988, one state was doing it. By 1992, 7 states were doing it. By 2003, only 11 states were using the EPA recommended indicator. It took some time, but the science and the data by themselves didn't accomplish change to the system, but ultimately the science was convincing and led to long-term science-based, data-based requirements that were demonstrated to be protective of public health. So now we get to agricultural water. Swimming isn't the same as irrigation. So why borrow those old E. coli numbers? One thing I neglected to say on this chart was that these sites, and we've discussed it a few times in the historical data, these sites were chosen because they were next to major sources of recent fecal contamination. So under conditions where you have a direct discharge of human source fecal contamination nearby the swimming beach, there is a demonstrated link. But when it comes to agricultural water and irrigation water, you don't necessarily have a city discharging raw sewage nearby. You don't necessarily ingest 100 milliliters per individual who is consuming. So the rules, the expectations behind those science made it a little bit more difficult to justify why are we using 126, 410, 576, because we don't know what the health outcome, we don't know what the risk is, because the situation, the sources of the contamination, the age of the contamination, and the route of ingestion are all dramatically different. But what doesn't change is these are just my words, and this is how I like to put it. If you have E. coli, you have poop. If you have poop, you might have fecal oral pathogens. And we know that many foodborne illness outbreaks are caused by fecal oral pathogens, and many recreational water illnesses are caused by fecal oral pathogens. It is a good idea to manage your water quality in relation to the fecal content and the chances that you might have fecal oral pathogens. No arguments there. It's just about the question is about what do those actual numbers mean. So if you want to see why FDA chose their numbers, I would recommend you look for this. This is in the list of resources that we're going to cite later in the document and that Linda has on the website. Samir Assar, who leads the produce safety program at FDA, did a Q&A about why did FDA use those numbers. LGMA has an appendix that I'll reference a little bit later that gives the scientific basis for their numbers. So I'll just recommend that you, if you're curious about why these numbers were chosen, go to the source and I'll let them explain in their own words. FDA is revising or reevaluating their decision about using 126 and 410, which is what came out of those EPA studies, because of the widespread concerns about the complexity of the requirements and the practicality of implementing them. So it's one way of voicing the concerns or responding to the concerns about do we know that these numbers are worth the effort to generate these numbers. Subpart E has requirements that are currently written but not currently enforced. One thing to encourage is that Subpart E requirements are based on good agricultural practices, and even though they're not requirements, they are informative, and those Subpart E requirements in produce safety rule can help a farm avoid adulterating produce. So while FDA reconsiders the standards in their documentation in support of the compliance date extension, FDA recommended that farms follow good agricultural practices to maintain water quality and develop water management strategies to identify and reduce risks. LGMA has a continuing effort to reduce risk from water, in part in response to the recurring outbreaks of E. coli 0157 based disease with a vehicle that is sometimes romaine lettuce, sometimes other leafy greens. LGMA has extensive documentation to assist farms with how to manage the quality of their water. As an example, 35 pages, more than a third of their metrics main document is devoted to water. They have Appendix A, B, and C, which all involve a substantial portion about agricultural water, and Appendix B is the technical basis document, which I referred to earlier. If you wanted to understand more about why LGMA made their decisions, that would be a good place to start looking. So here are some references and resources. I hope that this has been informative for you to understand why monitoring E. coli is such a good idea for understanding fecal contamination, but also why it's very difficult for us to throw out a number that we can stand by that says, this is okay, this is not okay, and why it's taking so much effort to reevaluate those. I do want to take the opportunity, since we're all talking today, to talk a little bit about inspections, in case anyone is curious about CDFA produce safety rule inspections. Just 2 minutes of information. We have inspectors that are in 5 different regions of the state that represent different commodities, different climates, different operating practices. Our inspectors work within their regions, but they also support efforts across regions. There are generally 2 types of inspection. One is an initial inspection that's going to be primarily educational. And then the follow-on inspection or a routine inspection might be more specific, looking for specific issues that ought to be resolved. A couple of things to make sure that you're aware of to take away some of the concerns about inspections that we hear about. They are announced. When possible, we will schedule the inspection with the farm. The time for an inspection varies. It might take a couple of hours. It might take up to a half a day. The inspections, unlike a GAPS audit or even participation in the LGMA program, a FSMA produce safety rule inspection is not optional. And then finally, the farms will receive a report from CDFA describing the findings. We have some information online to help you understand inspections, and we just updated, I think it was Monday, I think it was earlier this week, we published an updated version of Learn More About a Produce Safety Program Inspection that has a series of frequently asked questions that I think you will find informative if you have questions about the inspectional process. So I'm going to turn it over. I'll leave it up to Dr. Harris as to whether we need a break. And thank you very much for your attention.